Webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website, autism.org.
Now, before we get started, we'll introduce our speaker. Dr. Emily Casanova is an assistant professor in the neuroscience program at the Department of Psychological Sciences at Loyola University in New Orleans. Throughout her training and early career, Dr. Casanova's research has been focused on the study of autism from various perspectives, including investigation of its overlap with hereditary connective tissue disorders, such as Ehlers-Danlos syndromes and Fragile X permutation, as well as investigation of major effects of autism susceptibility genes and their evolution. These webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website at autism.org. And now I will turn it over to Emily.
All right. Thanks so much. It's really great to be here in the cyber universe. And hopefully this is a topic that you all are going to enjoy. It's definitely a little bit different than the topic I tend to talk about. I tend to get asked more about the Ehlers-Danlos stuff, which of course is of growing interest in the stakeholder community as well. But today I'm going to talk a little bit about some of my other work. For some years now, I've been studying some of the origins of autism susceptibility genes and kind of studying trends in those genes. So you can kind of see a bunch of weird little fish-like creatures in the background of my first slide. That may give you some hints of some of the stuff we might talk about today. But today is going to be probably a little bit more heady material than I tend to get into, but feel free to throw questions out. I'm going to try to break it down as much as possible so that hopefully everybody can kind of get some of the relevance of some of the data that we're going to be talking about today. And at the end, I'm also going to talk a little bit more about how that applies, so a little bit more slightly real world application. Okay. So first off, let me get my thing working. There we go. Okay. So you're going to see a bunch of fish and ancient pictures today. So definitely different than my Ehlers-Danlos stuff. So today, first off, I'm going to start talking about gene radiations over time. And by radiation of genes, I just simply mean the evolution of genes, how genes have arisen, because every gene has a birth, right, at some point in time. And so one of the things in my work that we've found is that there have been large radiations of genes, large groups of genes have evolved. And that happened over a certain period of time, but then it seemed to have dramatically slowed by the time we get to some animals known as tetrapods. And tetrapod, for any of those who, any of you who aren't already familiar with that term, which I assume is probably quite a few of you, that simply means that those are vertebrate animals, so basically fish, that started slowly coming onto land over time and eventually evolved into animals like ourselves. And this happened in a period called the Devonian, and that is about 385 to 360 million years ago. So it was quite a while ago that we finally wandered onto land. But we do see a lot of these genes evolving before that point. And so I've got this little histogram here, and it's arranged in kind of chronological order. So oldest period is towards the left, and then more of the newer clades of animals, the younger clades of animals, such as the apes, are towards the right. And you can see that the numbers of genes that are evolving over this time, we get a big burst early on, up until the point of around the bony fishes down here, and because not all fish had bones at one point. Some of them had more of a cartilage type skeleton, nothing that was really calcified. And so we see a lot of the genes that are evolving pretty rapidly over that time. But then it calms down once we get past that point. We get to some of the other types of fish, and then the tetrapods that we were talking about. So then gene evolution slows significantly, at least the evolution of new genes, right? No, we're not saying that gene evolution has stopped, right? But we're not necessarily inventing a whole bunch of new genes. All right. So here I've just highlighted in orange the period around which we see the early vertebrates. And so a vertebrate is anything with a cartilage or bony vertebral column that's wrapping around the spinal cord and protecting it. Also, you see the skull developing around this time too, and protecting the brain as well. And so that's basically how the vertebrates got their name. And vertebrates, the early vertebrates are synonymous with the early fishes. So the earliest fishes were also some of the earliest vertebrates. And so it's in these early fish that we see a major leap in the number of genes that are evolving during this time. And you can see that in orange here. And one of the things that we know that happened during that time, because we've used kind of a comparative genetic approach to study the genomes of different animals, we know that during that time of these early vertebrates, these early fish, that there were 2 rounds of whole genome duplication. And whole genome duplication is exactly what it sounds like. The genome, the entire genome, has been duplicated, and that happened twice. So that means that all of a sudden, these early animals, these early fish, have a huge amount of extra genetic material. Now, that does not mean that they keep all of those genes. A lot of those genes are lost over time just because they're not really necessary in extra copies. But then there's a subset of genes that do tend to be kept over time. And we're going to talk a little bit more about those guys and how they relate to autism and some other aspects of nervous system genes and developmental genes in general. So we see these whole genome duplication events and a large increase in numbers of genes all of a sudden in these early fish. We also see a bit of a peak too in some animals called the bilateral animals, and these are a little bit earlier. We're all bilateral animals ourselves, so you can take a line down our center and basically cut us in half and fold us over, and we're kind of mirror images of ourselves. So that's known as bilateral symmetry. A lot of animals that we're familiar with have bilateral symmetry. So here we've also got a little picture of a beetle. We have a flatworm, a planarian right there, and your dogs and your cats, all of us, we're all bilaterally symmetric. Before the bilateral animals could be symmetric, but they were more likely to have something called radial symmetry. And all that means is you could basically split them as if they were a pie, right? They have a radial form of symmetry. And some of the earliest animals didn't have any symmetry at all. But anyways, so we have these bilateral animals that have evolved, and there seems to be, again, a large increase in the number of genes that may have occurred around that time, and maybe also is involved in some of the evolution of bilateral animals, because there's some really interesting stuff happening to the nervous system during this period. Okay, so when we talk a little bit about something called gene conservation, we can look at gene conservation in a number of ways. And conservation just means it hasn't changed all that much, right? We might be talking about the conservation of the sequence of a particular gene, how well conserved that sequence might be. In this instance, we're going to talk about the conservation of the total number of genes that we have in our genome. And so again, on this graph, we're kind of looking from left to right. We're kind of going a little bit in chronological order, so earliest to most recent. And we can see that, especially in this very light blue line here, that's representing the tetrapods again that we were talking about. that there's not a lot of variability in the tetrapods in terms of the numbers of genes that we have. Whereas if you look at some of the other big animal groups, like in dark blue, we have the other fish, so the non-land vertebrates, right? And we also have in green, we have another group of animals that includes things like insects and crustaceans. And then even earlier in yellow, we have stuff like sponges and sea anemones and, some of those little crinoid flower looking animals that live in the ocean. And they all tend to have a lot greater variability in the total numbers of genes that they have in their genomes. So tetrapods are a little bit weird in that sense. So we're going to talk about other aspects of gene conservation also through this lecture. So so we do see some significant variability. And as I said, the tetrapods, we have a pretty minimal variability in terms of the total numbers of genes that we have in our genome compared to our cousins, the fish, which have some pretty significant variability. You can see, look at those spikes. They're pretty exaggerated. And just as a reminder, I think I already addressed this, but just in case I hadn't, we humans are tetrapods. Okay, so tetrapods are named for four-limbed animals, four-limbed vertebrates. Okay, and so we crawled up out of the oceans, and even though we stand on two legs, our ancestors are the tetrapods, so we can still be considered a tetrapod. So we would fall in that little light blue line in the graph that we saw before. Okay. Now, before we talk more about how autism genes have evolved over time, we're going to talk a little bit about some basic aspects of their structure and their function, just to get a little bit of background. So a while back, there was a group of scientists, and they had reported that a lot of autism genes were unusually long. All right. And they tended to be when you targeted a particular enzyme called TOP1 that regulates their expression patterns, that if you inhibited TOP1, then you actually inhibited autism genes in particular. So TOP1 seems to regulate the expression of genes that are very long, which happens to include a lot of autism genes. So this was an early indication that a lot of autism genes have kind of an unusual structure, and they tend to be quite long. So we went on to further study this. So we also looked at gene length, but we also looked at other things like the protein length. And we'll talk a little bit, we're going to do a little bit of genetics 101 before we get too far into this, so everybody's kind of on the same place. But we looked at protein length. We also look at something called transcript number. And what a transcript is a gene, when a gene is expressed and you produce something called RNA from that gene. So before you even make that into and translate it into a protein, it's kind of in an intermediate RNA stage. Okay. And that's called a transcript. And so each gene can have a lot of different versions of transcripts. Not every transcript is identical. They can sometimes do slightly different things. And so that is a really interesting aspect of complexity for genes in our genome. So we can look at the number of transcripts that a particular gene might make. And we can look at variations across whether that's an autism gene or that's a non-autism gene. So we do see some differences that we'll talk a little bit about. We also look at something called conserved noncoding elements and also transposable elements. Once we get to that information, I'll explain more about what those actually are, because I'm going to assume practically nobody here knows what in the world I'm talking about, and that's okay. And then finally, we're going to talk again a little bit about some other aspects of how autism genes may be conserved compared to other genes within the genome. So some different aspects of conservation. So those are kind of the big overview. So before we go on, though, I want to do a little bit of basic genetics 101. Okay, so we've got our gene, and this is my very bad PowerPoint version drawing of a gene, okay, down at the bottom. And so each gene has something called a promoter region towards the front, okay? And that it's not part of the coding sequence of a gene. It's not ultimately used to produce a protein, but it's a part of the gene that's regulatory. And so it helps to regulate whether, when, and how that gene is going to be expressed or not. And so we've got our little promoter region here in orange, and usually it's a binding site for other types of proteins that may start expression. It may repress the gene. There's a number of things that it can do. So we've got our little TFs, our little transcription factors, and those are just a protein, and they may form a little bit of a complex with each other and regulate gene expression somehow. Then in red, we've got our exons, and these ultimately are the sequences that are going to code for protein down the line. And then finally, we have our introns in blue, and these lie in between the exons. And ultimately, before you get to the final transcript, the final RNA transcript of that gene, they're spliced out. So the genome is really good at doing that. But why do I bring up introns if they're going to get spliced out? Well, sometimes there's some really interesting stuff tucked inside those introns that can affect how the gene is expressed or any number of things. So there can be some really cool regulatory features inside some of those introns that we'll talk a little bit about today. Okay, so as I said, we've got a transcript that's produced from the gene, and then that transcript ultimately, after you splice out the introns and you paste your exons back together, that ultimately is used in a process called translation, and that produces a protein product. Proteins are a major basic building block of the body, of cells of the body. And so not every gene produces a protein ultimately, but a lot of genes do. And those tend to be the ones also that we understand the best, because we can look at what their protein does in the cell. And it tells us a little bit more about what their basic function is. So in our study, some of the things that we can measure and investigate in terms of genes and proteins, we can look at their length, right? We can look at the length of the gene. We can look at the length of the protein. We can look at how conserved the sequences of the gene are over time. We can see how many other proteins might interact with that protein that gene ultimately makes. Okay, so we can see how many partners and how complicated those interactions can be. And then we can also estimate when a gene may have originally evolved. What's its overall age? It's not always as simple as that, but we try to get guesstimates, okay? So those are some of the things that I've done in my work in order to better understand what are these genes doing, when did they evolve, and so then pairing that with the time periods when they evolved, what were they probably doing in those early animals? Why were they important? Sometimes that can give us a better idea of what they're doing in our bodies today, rather than studying humans directly. Sometimes it's easier to study a fish, right? And get a better idea of what that protein at its most basic function might have been doing at one point or another. So that's kind of the general genetics 101. Hopefully we're all kind of on the same page. So let's get to some of the data. I know there's going to be a number of colorful graphs, so bear with me. Towards the end, we're going to be kind of pulling it down and talking about the different animal types and the different aspects of the nervous system that these actually relate to. So we're going to kind of pin it down, but in the meantime, just kind of bear with me. We're going to talk about some of these different features that I mentioned. So just like that earlier study that we talked about, we also found that autism genes tend to be unusually large. You can see autism in this whole presentation. We've got autism represented as the pink or the salmon color, okay? And gene length is represented on that y-axis going up the left side of the graph. And so you can see that the average line, this is a box plot, and you can see that the average line within the autism box is definitely much higher than all of these these other gene groups. So in the dark yellow, that's our controls. So that represents the genome as a whole. So what does the genome normally look like? What's it normally doing? And then we also have a couple of other groups that are interesting to pull out and compare to autism. So we've also got a developmental gene group in green. We've got a purple group that is representing nervous system genes, which you would expect we're going to see quite a bit of overlap with the autism group, right? And then finally, in blue, we have the immune genes. And immune genes tend to have their own very unique patterns because the immune system is rather unusual as far as a gene group. So they're interesting to compare against as well. But the ones we're going to focus mostly on today are going to be the autism ones in pink and then the controls in that dark yellow. All right, so we do see that these autism genes are unusually long. Then if we take a look at the protein lengths, we also see that the proteins in the autism group are unusually long too. Now, one of the things that suggests is like, well, Emily, why is that really important for a protein? Who cares if it's long? Well, longer proteins tend to have more domains, more functional things that they can do. So the longer a protein, that suggests that it may be doing some really interesting stuff, and sometimes maybe even a variety of different things compared to a shorter protein. So that means that autism proteins may actually be pretty complicated and have quite an interesting molecular life compared to the average gene in the genome. And so if we look at the relationship between gene length and protein length of genes across within the data set we're looking at, right? We can see on the left-hand side, we've got this little blue line here. It's forming what's called a sigmoidal curve. It's just an S curve, and that's exactly what it sounds like, kind of looks like an S, okay? And we can see that in general, there seems to be a positive relationship, right, between the length of a gene and the length of its protein. So as the length of the gene goes up, so does the protein, and vice versa. Now, if we pull out just the controls, because the ones to the graph to the left is all 5 of our groups smushed together, right? But we want to look at the controls in particular, right? We want to see what is the genome normally doing, all right? And so we can see that there's a much better agreement on the right-hand graph, right? So we can see that those genes, every time a gene gets bigger, the protein probably is going to get bigger. There's an agreement between those. So that's our control group. Now we pull out the autism, we look at these guys. And so there is a weak positive correlation between gene size and protein length, but we can see that once we kind of get up to, let's say down here on the x-axis down below on this right-hand graph, once we get to, let's say about 250,000 base pairs of gene length, that's a pretty big gene, okay? Once we get to that point, the protein length just kind of plateaus off. It doesn't really get much bigger. So these autism genes are getting gigantic compared to the relative size of the proteins that they're ultimately producing. So that's something that's a bit unique about this autism gene group. And you can probably imagine that this gene group, when we throw them all together, they're probably one of the major ones that is skewing that the left-hand graph here. So okay, so we went on also to study transposable elements and conserved non-coding element sequences. Now, these are the ones that I promised you I was going to tell you what in the world I was talking about, okay? So first off, transposable elements. These are small segments of DNA in the genome that are capable of either cutting and pasting or copy and pasting themselves around the genome. So they can make a lot of copies of themselves. Now, these are not necessarily genes in and of themselves. They're much smaller, okay? Sometimes they may be as small as just 300 nucleotides in length, which is actually quite small. What we find in the autism gene group, all right, is that we see a dramatic increase in the number of total numbers of transposable elements. Now, when we try to, because we know that the autism genes are unusually long, right? So you kind of expect, okay, well, just for their overall size, of course, they're going to have more transposable elements. And so you have to also do an analysis where you kind of normalize that number by the overall size of the gene, right? And so once we do that, we see that to the right here, the density of these transposable elements really isn't that much different. All right, so we get kind of excited initially. All right, there's a lot more transposable elements, but for the most part, it seems it's probably in part in relation to the size of the gene. And when we pull that out and we look at the number of transposable elements per gene and then the overall gene length, we can see, yeah, there's a pretty strong correlation there, right? It's almost a one correlation, which is insanely positive. Now, then we look at these conserved non-coding elements. And what are conserved non-coding elements? So let's pick it apart. So they're conserved, right? So these are sequences that we as humans share with some other animals, right? In this instance, we're sharing these with mice. This was how we were identifying these. So these are sequences that are shared with mice, okay? But they're non-coding, right? So they're not part of the coding sequence. They're not going to produce a protein. So instead, they're more likely part of the regulatory elements that we see in DNA that helps to regulate gene expression on some level. And so we've got a bunch of these non-coding or these conserved non-coding elements tucked inside the introns of genes, as well as in other regions, but we're looking strictly at the intronic ones. And so we looked at the differences of these conserved non-coding elements across the different gene groups. First off, we look at the absolute number, the total content, and we see, as you would probably expect, because autism genes are really long, we've got a lot of conserved non-coding elements tucked inside those introns. Then we go ahead and do the same thing we did before with the transposable elements, and we normalize that by the overall size. And we find, lo and behold, they're still really significantly different. Even for density, there's a lot of conserved non-coding sequences tucked inside autism genes. Well, you may be asking, well, what does that even mean? Why is that even relevant? Okay, so to break it down, we've got a lot of potential regulatory sequences tucked inside autism genes. That means that probably the complexity of how these genes are expressed and the things that they're capable of doing is significantly different from what we see in the genome as a whole. The level of complexity in autism genes is probably, it's extremely high compared to other genes that we see in the genome, which kind of goes along with the fact that autism is just very complex, right? And so there's a whole variety of potential regions within the gene where you might find mutations, and they may be doing a variety of things to the gene. Sometimes you might see them targeting the coding sequence and they change what type of protein is produced. That's a very simple version. But then other times, if they're hitting one of these conserved non-coding sequences within an intron of the gene, it may change the types of transcripts that can be made from that gene. So it's not necessarily changing the fact that the gene can be expressed per se, but maybe it's changing how it's expressed. And that in and of itself could be a susceptibility factor for autism. So again, as with all things with autism, it gets complicated, and we see it even at the genetic level. Autism genes are complex. Now, why did we talk about transposable elements if they weren't really significantly different, especially when we took gene size into consideration? Well, there's been a lot of research in the past that's shown that transposable elements are often borrowed and can become a conserved non-coding element. They can basically turn into a regulatory sequence for a gene. And so that suggests that even though the number of transposable elements within the introns of these autism genes may not be really different compared to what you would expect for the size of the gene, but they may actually be turned into regulatory sequences at a much higher rate than you would expect from the background genome. right? And so when we look at the total number of transposable elements versus the conserved non-coding elements, again, we're seeing a really high correlation, which suggests that there probably may be a good number of conserved non-coding elements in autism genes that were once transposable elements. They were once these mobile jumping genes, and they've added a layer of complexity to these autism genes that you just don't quite see in all the other gene groups. Okay, so moving on to talk about gene conservation. So we talked a little bit about conservation at the level of the number of proteins you might see within the genome as a whole, okay, and across different animals. But now we're going to talk about gene conservation in terms of the sequence, all right? So how conserved a given gene is in its sequence. And so we have a few ways that we can look at this. So we have one thing called an exact PLI score. You don't really need to know what that is, but all it means is that it's basically a rating as to how sensitive a gene is to a mutation. All right. And so when we look at some of these exact PLI scores, you can see that across the different groups, we've got some different patterns, and I'm going to talk about those as well. We've also got something called an ARVIS score, and that is very similar to an exact PLI score. So I'm just going to kind of leave it at that. We've got a couple ways that we can look and see how mutation sensitive is a particular gene. And then finally, we look at something called copy number variance. Now, some of you might be a little bit familiar with the term copy number variant. It's something that's been floating around the autism literature, especially as far as genetics, for a while. But a copy number variant is essentially a section of DNA. It might encompass a single gene. It might encompass a few genes. It depends on the size can vary a little bit. And either it's in its most simplistic form, it might be a duplication of that region. So you may have a gene that's duplicated, or a few genes that are duplicated, or it may be a deletion. And so you may have a deletion of one or several genes. And so those are known as copy number variants. And we have rare versions of those, ones that seem to occur very infrequently, probably because they're very detrimental. And then others that are common, and they're probably more common because they're not as harmful, right? So we have two versions of those. So we'll also talk about those as well. So first off, with the exact PLI scores, and again, this is an indication of how mutation sensitive a particular gene is. So we can see in the controls here, let's start with the controls. We've got more genes kind of hovering around the 0 mark than around the 1 mark. Now, with exact PLI, the higher the score, the more intolerant or the more sensitive a gene is. Okay, so the higher it gets, the closer it gets to 1, the more sensitive it is to any kind of mutation. So you can see that a large portion of the control genes are really not all that sensitive to variation. A small portion are, but most of them are kind of hovering around this lower end. Now, if we look at the autism genes, you can see it's the opposite trend. The majority of them are hovering around that 1 mark, which means that autism susceptibility genes are very sensitive to any kind of variation in their sequences. They do not like to change at all. Now, if we look at the ARVIS scores, and again, these are kind of giving a somewhat similar look into mutation sensitivity, only in this instance, the grid is kind of flipped. So the lower the number, the more insensitive they are, rather than the higher the number. So here we can see that the autism genes compared to the controls, they're definitely hovering lower in number on this graph. So again, once again, we've got these autism genes, and they're very sensitive to any kind of variation. Then finally, we've got those copy number variants that I talked to you about. And so essentially, we find rare and common copy number variants. Now, if we look at the genome as a whole across the human population, we're not talking about any kind of clinical population, okay, but just the human population as a whole. Rare copy number variants, as the name probably indicates, happen much less frequently than common ones, okay? But across both of those types of variations, we see that they tend to, they're less likely to include autism genes, which means that when a copy number variation happens, because they randomly happen very frequently, actually, in the genome, it's just most individuals who have a copy number variation, unfortunately, don't tend to make it beyond the embryonic period. They tend to be deadly, unfortunately. But when they do make it out of the embryonic period, we see that very infrequently do they actually include autism genes. So that suggests that when a copy number variation happens and it includes an autism gene, it's probably much less likely to make it to birth, unfortunately. They tend to be a little bit more deadly, a little bit more deleterious, as we would say. So now, again, this is not a clinical population, right? When we see copy number variants, because if we went to a genetics clinic, we would end up seeing probably a lot more autism genes in these copy number variants that are coming up. Why? Because we're looking at a clinical population where we already know that something is not quite working right. Right? So they may have some kind of physical malformation, or they may have intellectual disability, or any other number of things. So that's going to be different when you go to a genetics clinic. But when we look at the human population as a whole, when a copy number variation happens, it's much less likely to include an autism susceptibility gene. All right, so a lot of conservation here. And then we're going to take a look at one other element of conservation, which I didn't quite talk about yet. But we have something called an evolutionary rate, okay? And each gene gets its own rating, all right? So we can look at evolutionary rates of genes across animals in general. And so it's basically an indication of how much that gene has changed over time. And so some, genes that have higher ratings, that means they've changed a lot more across species over time, whereas ones that have lower rankings, they've changed considerably less, and they're what you would call more conserved. And so we do see that in the autism group compared to the controls, we do see that autism genes have lower evolutionary rates. So that means that across a large span of time, not just in the human genome, but we're talking, more than 500 million years now, that the conservation of these genes has been quite significant. They have not liked to change for a very long time. It's just not in humans. It's not just in tetrapods. It's for a very long time. So that's another interesting thing. So in general, we see that autism genes across in many different ways are very tightly conserved. They do not like to change. Now, one other way we can look at this also, it's a gene's sensitivity to change, is we can look at how the gene is inherited, right? And so if there's a clinical phenotype that we're interested in, like in this instance, we're looking at intellectual disability related genes, we can see here that there are different patterns of inheritance in terms of when that intellectual disability may be expressed. So we did this a number of years ago, and we had a large group of forms of intellectual disability that do have a single gene that's associated with those conditions. So we tried to simplify things. We didn't use anything that had like copy number variants or anything like that. And we broke these down further into two subgroups. One subgroup are forms of intellectual disability that did not have any association with autism. And then another group where we did have intellectual disability and it did tend to be associated with autism at somewhat high rates. Okay. And so that one is the blue group. And then the non-autism ID group is the one in pink. And so we can see that in the non-autism group, the pattern of inheritance tends to be overwhelmingly recessive. Now, what does it mean by recessive and dominant? All right. So for any of you who maybe vaguely recall your high school biology class, all right, if you did a little section on genetics, you probably did a Punnett square in terms of inheritance patterns. Maybe they use the example of brown eyes versus blue eyes, right? And brown eyes are supposed to be dominant. You only need to inherit one version of brown in order to have brown eyes. But if you want to have a blue-eyed child, then you have to have two blue-eyed versions, right, in order to actually express that phenotype. So it's not always that simple. I know it's really simplified in biology. It's not quite that simple in eye color patterns, but we'll give it that. Okay. So in this instance, we're talking about intellectual disability rather than the color of eyes. And so in the group to the left in pink, predominantly, you need to inherit two copies of a particular gene that are not quite working right in order to have these forms of intellectual disability. Now, when we look at the autism plus ID group, the pattern is a little bit reversed, not quite as extreme, but it's more reversed. And we see that about 60% of those different forms of intellectual disability and autism, they tend to be inherited in a dominant fashion. So that means that you only need one copy of that gene that's not quite working right in order for that phenotype to be expressed, that intellectual disability and autism to be expressed. So again, we're talking a little bit about not only inheritance patterns, but how conserved these are and how sensitive these genes are to change. So kind of on that note, I'm going to bring up a topic called dosage sensitivity. And so each gene kind of has its own version of dosage sensitivity. Some are more sensitive and others are less. Now, what do I mean by dosage sensitivity? That just means that a given gene is more or less sensitive to minor changes in expression of that gene. Okay, so genes that are really dosage sensitive, that are highly dosage sensitive, they are more sensitive to just minor changes in expression patterns of that gene. And you're going to see probably more likely some kind of clinical phenotype with mutations to those genes. Genes that are less dosage sensitive, you may end up needing multiple mutations or mutations across multiple copies of that gene in order to actually see anything clinical. So it really does vary. Now, if we talk about why genes might be dosage sensitive in the first place, some of the things that seem to lead to dosage sensitivity of a gene is the fact that the protein that it ultimately produces, that protein may be involved with other proteins in what we would call a complex, okay? Or it might also be involved in a really important signaling pathway. Those two things seem to be key to dosage sensitivity. So for a gene that is involved in a really important complex or pathway, it's more likely to be much more sensitive to changes in gene expression, in part because it's not so much about the absolute production, like the total production of that gene, it's about the relative ratios of that gene product to the other ones that are involved in that complex or that pathway. So if you change one, you've got to change them all if you're not going to see something bad happen. Okay, so just changing a single gene, even just a little bit, in some of these very sensitive complexes or pathways can affect the function of the entire complex or pathway. And so this is something that's known as the gene balance hypothesis. And so genes that produce products that are involved in these types of complexes or pathways tend to really weed out mutations very quickly, right? Because they tend to, more often, they're more likely to be deadly. Most of the time they happen, we don't actually end up seeing them because the embryo or fetus has been kind of self-aborted. But we do sometimes see it in infants who make it to past birth, right? So we also see, and before I go on, let me just switch maybe a little bit to the picture at the right to give you a better idea of how dosage sensitivity kind of works. So up at the top on in example A, we've got two genes and they're producing two proteins. One is called A, the other is called B. All right, so A is in blue, B is in red. And so A produces about twice as much. And in the end, they're forming a complex that's basically ABA. It's blue, red, blue. And it's able in this one to produce 4 complexes. Okay. And let's just say that that is the ideal number. That's what the body wants to use. Okay. Now, if we start changing A relative to the amount of B that's available. So in options B and C, we're doing that. So you can see that here in option B, you're making much less of this whole protein complex. And the same thing even for option C, even if you've got a lot more of B available, you're still only making like 3 full complexes compared to the original 4 that you made up in A. And so it's this relative ratio between these different component parts. In another way, think about it like you're having to put together a car, right? And you're always going to need a certain number of parts, and they're all going to function in a certain way. And if you have too much or too few of one, you're not going to make the allotted amount of cars that you need to make, right? You're always going to either be left over with parts, okay? And so let's say you need 10 cars in general, but you've got a gene mutation and you're only able to make 8 cars. It doesn't matter how many doors and chassis and hoods you may have left over, right? You need the full complement in order to have 10 cars. And that's what you need in order to live properly is 10 cars. All right. That's our metaphor for a protein complex is cars. So one of the things that we see with these genes that are really dosage sensitive, okay, is they don't like to be duplicated and they don't like to be deleted when it's single gene duplications or deletions. And that makes sense, right? Because we're talking about ratios. So this is why they're underrepresented in those copy number variants we talked about, as long as we're not looking in a genetics clinic, right? But if we're just looking at the human population in general. But one of the interesting things, now you remember when we talked about vertebrates and we were talking a little bit about whole genome duplications when the entire genome is duplicated, it's at that point that these genes are overrepresented in the whole genome duplications, in the genes that are retained at following whole genome duplication. Because again, as long as you duplicate all of the stuff that's involved in this complex, in making this car, then that's okay. All right. So we can handle whole genome duplication even better than a single gene duplication or deletion. So that's a little bit unexpected, but when we're talking about this idea of dosage sensitivity, it kind of makes sense. So if we look at another type of gene called a metabolic gene, and there's a lot of metabolic genes. They make up like about half of the human genome, the protein coding genome, right? And they're basically kind of like housekeeping genes. They're usually constantly active. They're doing basic stuff, basic cleanup, basic metabolism, all right? So highly necessary. But the interesting thing about these metabolic genes is they don't tend to form these types of really intricate complexes or pathways. And so when you have a mutation in one of those, it usually doesn't cause too much of a problem, right? And so it's only when you have 2 mutations, one in each copy of the gene, that you actually get a metabolic disorder most of the time. That's usually the pattern. And so they tend to be recessively inherited. So a lot of us carry recessive mutations for metabolic genes. If we look at, there's a large group of metabolic disorders called lysosomal storage diseases, if any of you have heard of those. And these guys are pretty nasty diseases. They tend to be progressive, and they're usually recessive. And so, but a lot of us actually carry mutations, single mutations. So about maybe about 15% of the population has a mutation for a lysosomal storage disease. And that's because it's highly unlikely that we're ever going to run across a mate who has a similar mutation in that gene, right? And so the rates of these metabolic diseases are pretty low, because we're usually, at least in the United States and in some other countries, we're usually not marrying close cousins, right? So that's how we reduce the rates of these recessive diseases. So anyways, I kind of get off a little bit of a tangent, but I think maybe for this group, that might be interesting information. So, okay, so if we look at those metabolic diseases, okay, so we see that they actually have a lot more single gene duplication and deletion. So you see them overrepresented actually in copy number variations, but then they're underrepresented in genes that are retained after a whole genome duplication, simply because it's not because, you know, it's bad to have an extra copy, it's just not really needed. And so if you lose a triplicate copy of one of these recessive genes, it's really no skin off your nose, right? It's not going to be too bad of a thing. So they do tend to get lost over time. But if you try to lose one of the extra dosage sensitive genes now after this whole genome duplication, that's when you're going to find problems. And that's why they don't tend to be lost after whole genome duplication. So you all are now experts now on molecular biology and dosage sensitivity genes. So congratulations. I know this is a little bit harder material than I tend to go through. Okay, so moving on. I'm also going to talk a little bit about protein-protein interactions, right? So again, talking about dosage sensitivity. And so we know that autism genes are all types of highly conserved, right? They don't like to mutate. But now the question is, okay, hey, we know they look kind of dosage sensitive, but are they having a lot of these protein complex interactions that we would expect? And so we find that, yes, actually, autism proteins, you know, are forming more relationships with other proteins. They're directly interacting and probably forming a lot more of these protein complexes. So that kind of goes along with our expectation, what we would expect to see with highly conserved dosage sensitive genes. Now, finally, we talked about age, because that's kind of how I started the whole thing off, right? And so we're going to look at gene ages across these different gene groups that we're interested in. And so we do find that if we look compared to the controls in yellow, that the autism genes are definitely older. So we see now we're going from oldest on towards the bottom of that y-axis up to the youngest, up towards the top. And the autism genes are definitely on average older. The immune genes are much younger, which kind of makes sense. And like I said, those immune genes have a really interesting, unique history in and of themselves. So again, yes, autism genes really old. And if we break it down and we look at the different time periods that a lot of these autism genes seem to evolve during, we see almost 30% arose during that early vertebrate evolution, those early fishes, right? We see another big group seems to have evolved by the time the bilateral organisms. So remember, those are the animals that you can kind of cut in half like ourselves and fold over like a mirror. So by the time those bilateral animals have evolved, we see another big chunk there. So more than 50% of autism genes have arisen by these two time periods, right? Which is pretty early. Okay. So we're talking 500 million years ago, right? This is quite a while. We also see some other representations of autism in some of these other groups. So the jawed fish, the single-celled eukaryotes. So those are not animals, but those are some of our closest single-celled relatives. And some of the chordates and bony fishes, but you don't need to know too much about those. But the big take home here is to see that more than half of autism-associated genes have evolved more than 500 million years ago. And that's huge. So we're talking foundational elements to nervous system evolution. Before we move on, because I am going to talk about what does that mean for the nervous system a little bit, I do want to point out that especially with these bilateral genes here down in peach, okay, The data that we were using from a website called Ensemble, they unfortunately don't have representations of species within some of the earliest animals that I have highlighted here in red, like some of the sponges and some of the jellyfish and things like that, stuff that doesn't have bilateral symmetry. So the bilateral animals are very well represented on this website, but we're kind of missing data. So one of the things I do want to point out when we see that such a large group of genes are shared across all the bilateral animals, it's possible that some of these genes evolved even earlier than that, simply because we're just missing data points there. So that's something that we're planning on working on in future and trying to whittle down, because there's a lot of major changes in how the nervous system has evolved across these different early animal groups. And it would be really nice to know, is there more of an association, say, in the jellyfish, right, and what's going on in their nervous system with autism genes, or is it something even older? Are we talking sponges who really didn't have a nervous system, but they kind of had nervous system-like elements that were evolving during that time? So that's a really important question, because I'm a really big believer in studying how things evolved to begin with in order to better understand what they're doing today. If you can understand the foundational elements, you're better going to understand what is actually happening in modern day bodies. All right. And so just to kind of bring that home again, there's just a lot of animal evolution, like a lot of the molecular components that we see in the nervous system, like different receptors and neurotransmitters, and even different aspects of how the neuron is shaped, because, you know, they look long and branchy, right? Even those things were evolving during this time. So there's still a lot of big questions. We're kind of scratching the surface here. We definitely know that a large portion of autism genes evolved during the early vertebrates. That is definitive. We have enough data points to conclude that. But earlier, it's a little iffy. All right. So we're hoping to get into that in future. And just to point this out again, so you can see it in a slightly different way. So we have a lot of those genes that are associated with those earliest fishes, those vertebrates. And that means that a lot of these autism genes most likely came from these whole genome duplications that we know happened back then. So those whole genome duplication events were a huge thing, not only for nervous system evolution in general, but also for autism nowadays. So that's a really interesting thing. And if we also look at these other two groups, so we also have nervous system and developmental genes, they have a very similar pattern like we're seeing with the autism genes here. So as you would expect, they have a lot of overlap. A lot of autism genes are nervous system genes, of course, right? Otherwise they wouldn't be associated with autism. So this is really interesting. We're continuing to work on this, but this is something that seems to typify not only the autism genes, but also nervous system and developmental genes in general. And if we look at that, so there's something called, okay, so we've got a new word for the day. I'm sure you guys have had lots of new words already, and I'm sorry to add one more onto you. But we've got something called an ohnolog. And what an ohnolog is, each gene is either an ohnolog or it's not. Okay. And what an ohnolog is that it simply means that that gene was derived, it evolved from a whole genome duplication. So you can imagine there's a lot of ohnologs that came out from that early vertebrate time period. And so the next question is, well, are there more ohnologs in the autism group compared to the controls? And so we can see that in pink, yes, definitely, we're seeing a lot more ohnologs in the autism genes than we see in the control groups. So again, our autism genes have a fairly unique evolutionary pattern. A lot of them have arisen from duplications, especially whole genome duplications. They have unique functions. They're very conserved. They're very dosage sensitive. They don't like to be mutated. And so this is a very interesting pattern that we see. The autism genes share a lot of these patterns with the nervous system and developmental genes in general. However, the autism genes tend to be more extreme when we look at a lot of these different features. So it's kind of like their nervous system and developmental genes, but on steroids. That's kind of how I like to think of them. All right, so kind of to bring it down and talk about some of the animals and some of the features of the nervous system that this might be relevant to. So we've got some modern representatives of those early vertebrate fishes that I talked about. And some of the modern ones, they're called hagfish and lampreys. They look a little bit more kind of eel-like. They have very long, slender bodies. They lack bottom jaws, so they're not really predators in the classic sense that they can go chasing something and bite it. Okay. They look a little bit more like sucker fish, to be honest, in some ways. But these were what sort of some of the earliest fish probably looked like. And so there's some features that we see in these fish that we don't see in some of their close relatives who are not vertebrates, all right, who are not fish. And so one of the main things that we see that developed strictly in the vertebrate lineage is that they developed what we would call a true brain. So before this, animals who were closely related to these vertebrates but weren't vertebrates themselves, they had what is known as a centralized nervous system, right? So it's kind of centering around the axis of the body. We have one today. We have a brain, we have a spinal cord, and then we've got the peripheral nervous system coming out into the periphery of the body, right? Now, that is not how early animals evolved. Many of the early animals that unfortunately are not represented in this data set, they do not have true brains. They do not have a brain. They may have a nervous system. They may have a centralized nervous system, but it's usually just a collection of neurons. But it's nothing that you would really put together and consider a brain. And so what we call a true brain, if we look at the fish diagram over here to the right, you can see kind of the circles, the colored circles in orange and green and blue. Those are representative, or those are kind of hovering over the forebrain, the midbrain, and the hindbrain. And so every true brain in the vertebrate lineage has three parts, and those are the three major parts. And you can see down in the diagram down below, even our brain has that today. Now, our forebrain is gigantic in orange, right? Because we're like, brilliant, of course. But then our midbrain is quite small, and then our hindbrain isn't too shabby. But we still have those same three segments of brain that the earliest fish had. That seem to have probably been influenced by those early molecular changes that we talked about. All right. So we've got more than a quarter of autism genes that arose during this transitional period when we're developing a true brain, a true three-part brain. And it seems to be probably a lot of that is due to the whole genome duplications that we had. Right? Because all of a sudden you have a huge extra amount of genetic material, and then you can start tweaking it to do slightly different things, and you may be able to expand and create new tissue types and just create a whole other level of complexity within the body, including within the nervous system. So these whole genome duplications are probably a major underlying factor, not only in autism nowadays, but even just in the development of the human type brain to begin with, or I should say the fish brain. We kind of have a fish brain. All right. Even earlier, we were talking about those bilateral organisms. And before that, that the nervous system was not centralized. Like I said, they kind of had nets of nerves around their body. All right. But they did not have a centralized nervous system. And so at least by that point, we see another quarter of autism genes that probably arose and may be involved in centralization of the nervous system. So there's some huge steps in nervous system evolution that are part and parcel of those time periods and may be associated with autism genes, which I find absolutely fascinating. We get to hear a lot about what's going wrong in autism-related genes, but I also like to think about what's going right and how it's part not only of the human condition, but just part of the animal condition in general, and what makes us so fascinating. So I'm going to finish off with a little bit of talk about functional enrichment, and all that is just talking about these autism genes and functionally what are they doing, how they might be different from control genes across time. And so we do see that up top we've got autism related in our little pie charts, okay, and we've got controls down at the bottom, and the sections in blue are representing the number of genes that are involved in the synapse of the neuron. Okay, so that's the little communicative ends with how neurons talk to each other. And so there are a lot of genes that evolved during these time periods, from the bilateral organisms all the way up to these early jawed fish. Okay, and we see that a lot of the synaptic components are evolving during this time period. If we look at how neurons project, right, because they have branching arms that project and connect to other cells. And so if we look at neural projections, we also see similarly a lot of genes over this time period that we're interested in are involved in neural projections. So we're making some massive strides in how neurons are probably connecting to each other during this time. And then finally, we also see a similar trend with neurogenesis, which is just the birth of neurons and how that happens and how that's regulated and the timing of things, right? So you can create new types of neurons for new different types of interesting tissues within this very complex brain that you're building. So we see all of those different elements are undergoing massive evolutionary changes from the time of the bilateral animals all the way up through some of the earliest fishes, which is just fascinating to think of how much the brain and the nervous system has changed and how quickly these things were evolving. We're talking, a couple 100 million years, if that. All right, so this is the time period we're talking about. We're looking at 750 million to maybe about 425 million years ago. Sounds like a lot, but also in evolutionary sense, it kind of goes by pretty quickly. So, but obviously a lot of stuff was happening during this time. So I just want to summarize all the things I've talked to you about today, because I know I've thrown a lot at you. So autism genes are unusually long. They tend to produce larger and potentially more complex proteins. They have more transposable element and conserved noncoding elements in their introns, so probably a lot of regulatory sequences. They have a greater density of those conserved noncoding elements too. They're very variation intolerant. On average, they tend to be older than the rest of the genome. They have a lot more direct protein interactions. They're highly dosage sensitive. They tend to be very tightly conserved across the animal kingdom, and they're often inherited in a dominant fashion. So if there's a mutation, you're probably going to see something going wrong. They radiated during very early animal evolution. Many seem to probably be the result of whole genome duplications, and they may have played really important roles in key steps in the evolution of the nervous system. And then finally, when we look at their functional enrichment, especially during these early time periods, we see a lot of involvement with the synapse and neural projections and neurogenesis in general. And so these are really going through pretty rapid evolution at that time. So these are references if you care to screenshot, if you're interested in any of those. And thank you so much for listening. Hope it wasn't too difficult. I enjoy torturing my students with this material too. So you get it for free. You don't have to pay tuition.
Well, Thank you so much, Emily. Thanks for that presentation. We do have a lot of questions and not a ton of time, but we will get to a few if you've got time to.
And I always have my email, which if you want to provide them, either my Science Over a Cup of Gmail or my Loino email, either is fine. And they're always welcome to contact me with if they don't get their questions answered.
Okay, great. I'll put that on the site so that people can email you if they need to. I'll get a couple in here though. One question was about, is there a reason why the exact PLI scores aren't more uniform in distribution?
That is a really good question. And I've thought about that myself, because it's an unusual trend that you see. Now, I will say I'm not an expert in exact PLI scores and how they're determined. But I think if we think about it from a dosage sensitive perspective, it's probably a little bit more of an on-off rather than a gradient kind of thing. So you're probably going to get more of, okay, this gene is definitely dosage sensitive, or this gene is really not that dosage sensitive. And I don't think you're going to get a whole lot in between for whatever reasons. I mean, either the pathway or complex is really important, or it's just more amenable to change. So that's at least the way that I've kind of thought about it. There may well be a different reason though, because again, I'm not an expert on that. And there may be something in the literature too that's maybe something that's already been identified before too. So
this next question is about the increase in autism. So we're seeing this sort of exponential increase depending on what data you're looking at. Would there be environmental triggers that might be part of that, or do you have any insight about that?
So just from my, slightly educated perspective, I can see it being highly variable across time, in part because a lot of these pathways are interactive, right? And they're overlapping, a lot of growth related, a lot of developmental related pathways. And so in some way, these pathways are almost a little bit designed to be plastic, right? They're designed to respond to environmental cues in a way, okay? Especially if you start talking about immune system and hormones and all of that. Those are designed to be environmentally responsive. And so I think some of what we're seeing is probably a reflection of that plasticity. It may not be the highest we've ever seen it. It's just we've only really recognized autism in the 20th century. I personally have to presume, like if you were to go back to, let's say, 19th century London, okay, when food was horrible, it was doctored, it was highly poisonous, the air was poisonous. I have to assume that probably intellectual disability and autism were even at much higher rates in a situation like that. But again, it's designed to be plastic within a certain reason, right? But then also, we could be seeing some changes in how we're finding partners. Just think back in the day, we were 100 plus years ago, we were more likely to find partners who we were probably a little bit more closely related to. So on that hand, we're more likely to have recessive disorders, but there are other ways in which the genome kind of adapts to variation, and sometimes it accommodates variation. So if you start also then pairing with people you're not terribly closely related to, on the one hand, that's a good thing. But on the other hand, you may be starting to break up variations that kind of have adapted for each other. And you may start seeing some deleterious nature of some of those because you've broken up pairs that have kind of co-adapted. So I'm not saying that means we shouldn't marry across races and stuff. That's totally not what I'm saying. However, because there's always going to be risk, no matter if you're having a kid, there's always going to be a risk, right? It's genetics. It can be a crapshoot sometimes. But you may change the type of genetic variations you're seing in neurodevelopmental conditions. So I mean, recessive disorders aren't good either. So so it may also be some of the patterns we're seeing in moving around a lot And, I mean, that's okay, but it might be having some effect too. I don't think it's probably a huge one, but it might be measurable. So, yeah.
This is a comment that I think you'll appreciate. This person says, interesting to hear your research interests in both autism and EDS.
Mhm.
My mom was diagnosed a few years ago with EDS, and I have many of the symptoms too, along with MCAS symptoms.
Mhm.
Both of us are diagnosed as autistic. So
Yep.
I know you've
Yeah.
done quite a bit of work in that area, and we have a webinar that
Yeah,
you may wanna check out.
definitely. Yeah, if you haven't seen that, and you're always welcome to approach me too. I myself am a zebra, and so I take that part of my research very, very seriously as well. And I'm also a mast cell sufferer too. So, but again, going back to the idea of plasticity and immune involvement, we might be seeing more autism in the zebra community, in part because of that immune involvement. At least that's kind of one of my leading interests. So.
Okay, and this person's very curious about how some of the features of autism as expressed in humans may have very valuable features for other mammals and other animals. Have you considered that?
Yeah, I mean, it depends on where, how distantly related we are. I mean, there's certainly plenty of people who are doing autism studies on zebrafish, right? So, it just depends on what level you're looking at, right? And it's easier to study in species that are more social, where you might be able to measure anxiety levels and things like that, or at least indirect indications of anxiety. But yeah, I mean, in general, if you're talking about, a little bit less about, autism associated challenges, but just brain evolution in general and how these autism related genes are functioning in other animals. So one, they're probably doing a lot of similar stuff because remember, they're very conserved, right? So once evolution kind of figured out what to do with them, it probably hasn't changed it a huge amount, right? Because it's kind of one of these basic, it's one of these basic modules. Once you've invented the Lego, you don't want to reinvent the Lego too much, right? You can, mold it and change it in different ways and put it into different, different buildings and different structures. But once you've invented the Lego, you kind of stick with the Lego, if that kind of makes sense. But it's fascinating. I will say that the evolution of the nervous system is usually underappreciated in terms of how variable it can be. When you look at even just the overall structure of different animals' brains, we kind of tend to think of them as human but smaller, and that is so not how it works. There's an exceptional variability across different animals, which I get to teach in my human brain evolution course, which is always fun. And it's very underappreciated. So if you were actually to look at brains across many different animal clades, you would start seeing, wow, the different shapes. Even just looking at a dolphin brain can be insane. The way it functions, right? Because it has a tiny corpus callosum that connects between the two hemispheres, and that's so one brain can be asleep while the other one can be awake, so that it can surface to the water and still breathe. And so it kind of has two brains, which is kind of weird. So that's kind of an unusual one, but yeah, there's an exceptional amount of phenomenal and gorgeous variability in nervous system evolution, and it's all about what does the body need. I mean, that's the point of the brain, fundamentally, is to make the body move. How do you sense the stimuli? Then what do you do in response to that? And that varies by what the animal does, So yeah, it's cool. It's a cool area to study.
Well, thank you so much, Emily. Thanks for this great presentation today. We are looking forward to having you back soon.
Always.